Hi, my name is Marc Hoffman. I'm the director of the lymphoma program at the University of Kansas. I'm here today to talk a little bit about the abstract that I presented, which was golcadomide plus RCHP and polatuzumab vedotin and the treatment of therapy-naive diffuse large B-cell lymphoma. So golcadomide is a unique drug. It's a first-in-class agent of a group of drugs called CELMoDs. And the drug, what it does is that it binds specifically to two proteins, Icarus and Alos. It leads to their closed conformation. It binds outside of the pocket of the more traditional IMiD, which is the tryptophan pocket, and leads to rapid protein degradation. And so we saw the rapid protein degradation early on in the cell line data that was significantly greater than that of lenalidomide. We had nearly 100% degradation in all of the cell line profiles. And then golcadomide was specifically designed in order to distribute in lymphoid organs. And so it preferentially distributes in the spleen and lymph node compartments rather than the bone marrow, which is in contrast to the IMiDs that had been there previously. And so between that and the fact that we saw efficacy of the compound in cell lines that was independent of cell of origin, the study went forward to combine golcadomide with anthracycline-based chemoimmunotherapy in the frontline setting. So the data I presented today are actually the extension of a much longer study that included an initial arm of golcadomide plus R-CHOP, and then this arm of golcadomide plus RCHP polatuzumab vedotin. And the intention of the arm was to develop new data and to ensure that we had a similar risk profile and safety and tolerability analysis compared to that of the R-CHOP arm. And what we saw was that somewhat remarkably, in spite of there being more cytopenias in the polatuzumab arm compared to the vincristine arm in Pola-RCHP, we did not see significant increases in the cytopenias, and the toxicities remained manageable. The main on-target effect of the drug is neutropenia that you see in patients. And so the dosing schedule by day 7, the vast majority of patients do become neutropenic. This importantly did not result in any compromise in the delivery of the chemoimmunotherapy backbone. Those patients did recover by day 1 of the subsequent cycles, and only one patient out of the 50 that were treated in the 0.4 milligram cohort had to discontinue secondary to neutropenia. So the drug was well tolerated, and then in terms of efficacy, we had a CR rate of 89% and a 12-month PFS of 96% that was observed with the combination, which certainly compares favorably compared to the standard of care. The experimental outcomes of MRD also showed increased rates of undetectable MRD, particularly among patients who had high-risk disease, who appeared to have very similar outcomes to the general population. So in summary, it's a novel first-in-class agent that leads to more rapid and significant degradation of Icarus and Alos, leading to immune activation and direct cell killing in large cell lymphoma, and in combination with both R-CHOP and RCHP polatuzumab vedotin was safe, tolerable, and did show early signs of efficacy.